Assalamualaikum and good afternoon. So today we are going to proceed with our lab class where we will look at sample mounting and sample staining. So if we look at the previous lab, we've learned about the process of sterilization and the process of making a media. So today we are going to learn how to mount and stain a microorganism sample. Okay, so basically mounting and staining is used for viewing microorganism. So if you remember under 5I, okay, mounting and staining will go under the identification phase or identification part, where we will learn how or what is the techniques that we can use to make microorganism clearer when we view under a microscope. Okay, okay, so sample mounting and staining. Alright, mounting is the act of placing a microbiological sample on a microscope slide. Okay, so if you guys still remember, I told you that normally, when we culture microorganism, it's either inside of a liquid media or a solid media in the form of an agar. So, to be able for you guys to view the microorganism, you cannot put the media under the microscope. You must first place the microorganism from the media on top of a microscope slide. So placing the microorganism sample on top of the slide is known as mounting. Okay, so the purpose is to view the microorganism sample, and there are two main type of mounts. There's two ways that you can mount a sample of microorganism on top of a microscope slide. The first method is the wet mount, and the second method is heat fixing. Okay, and these two methods are mainly used in the lab when you want to view a microorganism on top of a microscope slide. Okay, let's look at the first part, which is wet mount. Okay, samples are placed on suspension liquid. Okay, so normally we use wet mount if the sample that you want to view, okay, is from a agar or a solid media. For example, if you culture a microorganism from a solid media on top of solid media, so if you want to view that microorganism, normally we will use the method of wet mount. Okay, so wet mount is suspension used as sterile liquid free of compounds that can distract view under microscope. Okay, examples: distilled water, double distilled water, any form of purified ethical grade water. Okay, so imagine when you culture a microorganism on top of an agar, normally that microorganism is dry. Okay, it is dry. So if you still remember the colonies on top of a microbiology agar, okay, when we use the inoculation loop, we will scrape the colony using the inoculation loop, okay, and then we will place the colony on top of the inoculation loop, okay, on top of a water which you have dropped on top of a slide. So the water that you use as the wet mount cannot be compounds or liquids which has a lot of uh, precipitation or a lot of mineralization or any type of distraction, okay, that will hinder your view of the microscope when you want to view the microorganism. So normally the water that we use for wet mount must be water free of any impurities, which is distilled water, double distilled water, or any form of purified analytical grade water. Okay, so wet mounts are normally used to sample required to be alive or starting step to heat fixing of solid media be sampled. Okay, so normally when you place the microorganism from your agar media on top of the wet mount, okay, the microorganism will be alive. Okay, it will be alive. So you can see if the microorganism can move, you can see the movement of the microorganism. Okay, so this is an example of a wet mount. All right, first you take a clean slide. Okay, you sterilize the slide using ethanol, and then you take a micropipette, a dropper, or a dropper, and you place one to two drops of water on top of the slide. Okay, so when you place one or two drops of water on top of the slide, now you have water drops on top of the slide. Okay, then you take an inoculation loop, you sterilize the inoculation loop, and you scrape. Okay, you scrape the loop, okay, to the colony of microorganism on top of an agar or a solid media, and then you place the colony from the scrape loop, and you mix the colony within the water. Okay, within the water. Now you have, as you can see on the third picture, you have the colony which is mixed with water, and then you put the cover slide on top of the sample and you can view it under a microscope. Okay, that's the simplest form of mounting, which is wet mount. Okay, just place a drop of water, take the inoculating loop, sterilize it, and take the colony from the microbiology media. Then you place the colony inside of the water and place a slide, and you can view it under a microscope. Okay, the second type of mounting is called heat fixing. Okay, where samples are fixed to slide using heat. Okay, liquid sample must be spread thin on slide before heat fix. Okay, so before you can do heat fixing, normally we will have to use wet mount first. Okay, we will see this after in the procedure. Okay, for samples for solid media, the same procedure is followed with wet mount, but an additional step of drying and fixing with flame. Okay, so for heat fixing, if the sample comes from a microbiology media, which is solid media, you'll have to undergo wet mount first before you can do heat fixing. But if the sample is from a liquid media, you can straight away take the liquid. Okay, straight away take the liquid which contains the microorganism and put it on top of a glass slide to undergo heat fixing. We will look at the procedure of heat fixing in the next slide, right? Okay, so it's easier to view to the dry condition. So basically, if you compare heat fixing with wet mount, heat fixing is easier to view under a microscope. Okay, and samples are normally killed by the flame, and motility cannot be viewed using this mount. So when you undergo heat fixing, normally your sample will die. Why? Because you have flamed the sample. Okay, now let's look at um, the example of heat fixing. Okay, first you take an inoculation loop, okay, and you spread the culture thin on, okay, you spread the culture thinly on the microscope slide, all right, on the microscope slide. So after that, you will have to allow the sample to dry. Okay, allow the sample to dry. So how do you know it's fully dried? It means that when it's fully dried, you will not see any more liquid on top of the slide. So the sample will dry and most of the bacteria or the microorganism will stick on top of the slide. After drying, you take the sample and you pass the slide through a Bunsen burner or a flame. So when you pass the slide containing the dried sample, this is when you are conducting, or this is what is called the heat fixing. You use the heat to fix the microorganism on the slide. So basically, when it's dried and you undergo heat fixing, the microorganism will be stuck on the slide. Okay, and a good example, if you still remember, when you view prepared slide under the microscope, normally when you view prepared slide, the prepared slide is created by using heat fixing. After that, you can stain it and view it under a microscope, either using normal lens or using oil immersion lens. Okay, that is the first part. Okay, that's for the first part for mounting. Okay, we will continue on the second part, which is staining. Part of our practical class, okay, which is staining. Okay, so if in the previous lecture we've learned on how do we put a microbiology sample on top of a slide. Now we will learn what type of stains that we can, what type of dyes that we can use to stain the microorganism sample so that when we view it under the microscope, it will be much easier to detect and it can basically highlight the physical structure of the microorganism. Okay, so staining is basically a practice. Okay, it's a practice or an act of dyeing samples to make it easier to view with a microscope. Okay, so you are giving the either you are giving the microorganism sample color or you are coloring the background of the slide depending on what type of stain that we use. Okay, so there's a lot of different category of staining, but the most used is simple staining, negative staining, acid plus staining, and gram staining. Okay, so in this lecture or in this practical brief, we will look at the usage of simple staining, negative staining, and acid fast staining. For gram staining, we will be learning it on the next class. Okay, so the most widely used form of staining and the easiest form of staining to do is the simple staining. Okay,
The simple staining is we are using dyes which are positively charged, so that when we dye the samples, normally the microorganism sample are negatively charged. Okay, so normally when it's negatively charged met with a positively charged dye, it will be easier for the dye to enter the microorganism, because positive and negative will bind together, and when they enter the microorganism will be colored. As you can see, an example is in this picture. Okay, the positively charged is the basic stain, and the specimen or the microorganism sample is negatively charged. So normally when we use simple staining, we will heat fix. Okay, we will heat fix the sample first. Okay, so if you still remember, we had two types of mount: the wet mount and heat fixing. So for simple staining, we will do heat fixing. Why? Because we want to make sure that the microorganism is stuck or is fixed on the slide. And the second thing is that when we do heat fixing, the microorganism will die. And when the microorganism dies, it'll be easier for the stain to enter the membrane of the microorganism. Because if the microorganism is still alive, okay, they can maintain the membrane structure, and they will not allow any stain to enter into their body. Okay, so the dyes that are mostly used in simple staining are methylene blue, safranin, and crystal violet. Okay, these are all positively charged dyes or positively charged stain. So simple staining is basically quite easy to do. Okay, as you can see in the procedure. Okay, we begin with heat fix the emulsion, or basically we heat fix the sample. After we heat fix the sample, we smear the preferred stain. It can be methylene blue, it can be safranin, or it can be crystal violet. And after we, so it's not just called smearing, it's also called flooding. Okay, because we flood the whole area with the preferred stain. And normally we let the stain settle within the microorganism for about 50 to 60 seconds. And after that waiting period, okay, we will use distilled water, and we'll wash away the stain in this manner. Okay, we have to slant the slide a bit. Okay, make the slide slant a bit, so that when we wash with distilled water, the water will flow into either a staining cup or a staining rack. Okay, so we cannot just simply wash away the stain into the sink, because the stain is dyes which are considered as hazardous waste. Okay, and when we wash the sample that has been stained, we cannot wash directly towards the sample. If you can see the picture above, the sample is in the middle, and after you stain it, we wash it from the top. Let the water flow and carry away the stain. We cannot wash directly to the sample, because if we do that, we might risk